Hello everyone. Today I'll be presenting a computational design for Prefusion-Stabilizing mutations in alphaviruses. My name is Mudit, I'm part of the Myo Lab. Let's get into things. So we're looking specifically today at VIV and MAV, and let's get into what alphaviruses really are. They're mosquito-carried viruses. They come in two different flavors, encephalectic, which encompasses VIV, and we can see neural inflammation as a major category of it, arthritogenic, which encompasses MAV, and this is basically characterized by joint inflammation. Now, when people ask me what is so special about alphaviruses, I like to tell them that VIV was actually weaponized by the USSR as an aerosol bioweapon. But some more modern pandemic drivers are the Aedes range, which is the mosquito's habitats, are increasing with the warming climates. There aren't any licensed vaccines or antivirals for VIV and MAV. And when an outbreak occurs, which happens in many third world countries, over 75,000 people are impacted by said outbreak. So our goal overall is to create a vaccine for this problem. And so, we know that alphaviruses are envelope viruses, and antibodies target envelope proteins. So when we look at this image of an alphavirus right here, an antibody would be targeting this portion, which makes it extremely important for us to stabilize that zoomed in. And that's our goal for this computational pipeline. We want to stabilize the E1, E2, E3 prefusion complex. Now you might be asking, why is stabilizing this complex important? And the answer is quite simple. It improves the shelf life, the solubility, the expression, as well as just the stability of the entire vaccine as a whole. With that being said, let's move into the actual pipeline. Now there are two portions to this pipeline. One is the computational portion, which is all automated, and the other is the manual portion, where I myself go through all the mutations. So we start out with a experimental PDB, that's just the alphavirus. We refine that PDB via Cartesian relaxing, and then we apply a number of methods, thermo-MPNN, protein-MPNN, etc., and we essentially gain 5,000 mutations. With these 5,000 mutations, in VIV specifically, 477 were unique. We structurally validated to check if the mutations actually made sense, and we had about 160. From there was the manual steps, which is what I'm going to explain in the rest of this presentation, where we essentially go from 160 down to 24. So this is just a basic idea of the 160 mutations. VIV is a length of 869 residues, and there's 99 positions highlighted in cyan here, and these cyan are the hypotized mutations. We have this first shortlist criteria where we remove these surface mutations. As we know, antibodies do target the surface of a protein. So if we mutate the surface, then our vaccine is pretty much ineffective, because if we're mutating the surface, it's not going to be like the wild type, and that's why we've eliminated all these surface residues. So as you can see here, they're just a comparison image. The surface residue is clearly interacting with the solvent, hence we've eliminated it. However, the non-surface residue is pretty packed in the molecule, so it will not interact with the solvent, so the antibody will not be detecting it, and hence we kept it. We've also removed all the ones that have a decreased number of hydrogen bonds. Hydrogen bonds stabilize the mutation as well as the entire molecule. If we remove something that has a hydrogen bond, not only are we impacting that mutation, also the other things that are binding to the hydrogen bond, they don't have something to hydrogen bond to. So that makes the entire energy actually unfavorable. And so we've removed everything that has less hydrogen bonds, and we've kept everything that has the same number of hydrogen bonds or more hydrogen bonds. And so, with all of that, we have came up with these results for VIV, 24 results for each one. And so I just like to appreciate the fact that we've came from 5,000 computational, we filtered it down to 477, 160, and then 24 mutations. An important thing to notice also is that there's about a 49.3% complementarity between VIV and MAV. And what's important about that is when we make mutations, some of them are actually the same at the same spots. So these highlighted in yellow is actually similar mutations. And so what we are doing in a vaccine is we can multitask and we can use one mutation for multiple vaccines. So this is just a basic image of where our final mutations sort of line up. This is how everything looks shortlisted, and here's the same for MAV. Now, let's just take a quick recap. We've basically created a computational pipeline that takes a sequence, converts it to PDB, generates mutations that we can actually go into lab and test. And so we've already applied this to VIV and MAV, as you've seen in my presentation. Our next steps for this are to increase this to the rest of the alphaviruses, and test these mutations in a more lab setting. Anyways, thank you for listening. I'd like to acknowledge my mentors, Christina, Rocco, as well as the MyoLab as a whole. If you have any questions, feel free to contact me on my email. Thank you.